Okay, gang, so the video I have posted already on neurons and neuroglial cells that is useful for you to look at, but I'm going to do a more comprehensive video here over Chapter 12. So please feel free to watch both, and some of the material will be repeated. So again, this is Bio 207 hybrid with Dr. Granier at the Meramec Campus. Chapter 12, which is our neuro block. This is going to be our final test, this chapter, along with the material all the way through Chapter 17. So the big thing we need to go back to is in Chapter 4, we talked about the 4 tissue types, and nervous system was one of the 4 tissue types. And the way that a nervous tissue is different from a connective tissue, from a muscle tissue, is that the nervous tissue is going to conduct electrical impulses. And those electrical impulses are conducted upon a special cell, the neuron. All right, and so anywhere that we have neurons in our body, that is going to be an area where we are going to have some nervous tissue, and that nervous tissue might be embedded in the skin. And so we kind of did talk about some of the neural components of the skin in Chapter 5. And then we have talked a little bit about the neurons controlling the muscle in our skeletal muscle system. And so we're just going to go back into a broader sense of where all of the neurons and the supporting neuroglial cells are located, how neurons work, how the neuroglial cells specialize into their jobs, and then we'll go back into, again, how the nervous system then is divided into the different areas and groupings based on functionality and anatomical location. Okay? So when we look at the nervous system, you need to kind of be able to make a flow chart and understand all the key terms of that flow chart. So anything that has neurons, nervous tissue, neuroglial cells is considered the nervous system. So the organ system is where the neurons are gathered in large numbers and amounts in the brain and the spinal cord, and that grouping of those 2 organs is your central nervous system. But there has to be connections of the axons and the neurons in the brain and spinal cord to receptors in the periphery. And so your peripheral nervous system is going to be all of the nervous tissue, so the neurons and the neuroglial cells that enter and leave the brain and spinal cord, giving us our 12 cranial nerve pairs and our spinal nerve pairs. Now, some information is going to muscle, to glands, and trying to have an effect because neurons in the brain and spinal cord want to move this tissue or adjust the diameter of this vessel. And that information is part of your efferent, for exiting, affecting system. And so that it tends to be known in the peripheral nervous system as your efferent nervous system is your somatic nervous system and your autonomic nervous system. And they are going out to have effects. Now, some of your nerves of your brain are going to have efferent, so all of the axons coming out of that nerve might have some ability or be strictly abilities to go to nerves, go to muscles, go to glands, and have effects. The other arm of the peripheral nervous system is an afferent nervous system. So it's the system where all of the information is coming from a receptor. That could be a taste, a temperature, a pain, a photo, an audio or balance receptor, a proprioceptor, a stretch receptor, and it's going to the central nervous system. So afferents are going to the central nervous system. So all of the neurons that carry information from some type of receptor or carry information because the nerve endings or the sensory knobs of the nerve are embedded in a specific area of the body and carry information to the nervous system are going to be your afferent. Most of your peripheral and cranial nerves have mixing of some axons in the nerve are going out to have effects, and other axons that are part of that nerve, that are part of that gaggle of neuron axons combined into a neuron, to a nerve, are going to be afferent. A few of your cranial nerves are strictly afferent nerves. So olfactory nerve, the first nerve, is all of the axons that tie into the smell receptors. And so that olfactory nerve is almost strictly an afferent peripheral nerve that brings only information to the brain from the smell receptors, okay? So that's kind of one of those parts, all right? So when we look at the peripheral nerves, most of your cranial nerves are going to be sensory only, efferent, motor, gland control only, or they'll be mixed. Most of your peripheral nerves of the spinal orientation, so coming out of the spinal cord, are going to be mixed nerves. So they're going to carry both sensory information to the brain and spinal cord from receptors somewhere at that level of the spine, and then they'll carry motor and glandular outputs from that level, okay? Now, when we start looking at the efferents and we start looking at the control of motor and glands, some of our muscles are under voluntary control. They're skeletal muscles. So if we take those neurons that are basically motor neurons controlling skeletal muscle cells that are under voluntary control from the motor areas of our brain, those neurons and that collection and gaggle can all be put together as your somatic motor nervous system. Okay, and so that's going to be everything that is controlled that has some voluntary conscious component. All of the neurons that control gland and smooth muscles and muscles that are under involuntary control, so I don't think about it, I don't consciously am aware of the controls that are occurring to those muscle and glands, are going to be part of the autonomic nervous system. Now, when I want to control things voluntarily, I can control the number of motor units, and therefore I can control how much contraction is taking place from a muscle. So do I get 10% contraction, so I just lift a paper, versus 80% contraction, so I can lift like a big heavy rock. So I modulate some of that control of how much do I want force production-wise versus how many motor units I recruit. On the autonomic side, there's no conscious awareness. So in order to kind of get the right amount of force production, I have basically two opposing systems. I have the parasympathetic and the sympathetic system, and I kind of think of it as like a brake and an accelerator. I'm kind of learning how to push the brake and push the accelerator so I get the right amount of constriction or secretions that I need for that particular organ. So thinking about the heart rate, if I had an all or none response, like the motor units have, I would need to potentially control the heart by either I turn it on and I get a certain amount of beats, or I turn it off. And so I don't always necessarily have the ability to modulate my heart rate to 110 beats or 120 beats or 130 beats. It would be just all or off. So what I get when I get parasympathetic, sympathetic control is as I need to up my heart rate for a few beats, I can just push the accelerator. But then as I need to come down in my heart rate, I can take my foot off the accelerator and push my brake and bring my heart rate down. So it gives me that different level of control because, again, I'm not controlling it by motor units and recruitment. I'm controlling it by output of a little brake or a little accelerator. Okay. So the takeaway in this is, yes, we need to know that anywhere we have neurons, we potentially have nervous tissue. So if there are neurons in the muscle, that means there is nervous tissue in the muscle. If there's neurons and neuroreceptors in the skin, we have a neural component, neural tissue in the skin. When we start looking at the collections of neural tissue together, we see that they form organs. And those organs are the brain and the spinal cord, and those are housed in bone, so that's the central, whereas all of the big gaggle, big collections of axons entering and leaving the brain and spinal cord form our peripheral nerves. For the peripheral nerves, some of the axons entering are carrying only sensory information too, so that's why they are known as the afferent sensory part of the system, whereas some of the axons leaving are carrying efferent signals, and they can be divided into the efferent signals going to motor and under voluntary control, so the somatic motor system or the autonomic motor system. And because the autonomics has a brake and accelerator, you have the parasympathetic and the sympathetic arm. Okay? So again, just getting comfortable with the terms, the CNS, PNS, ANS, motor efferent, sensory afferent, sympathetic, parasympathetic, somatic. You just need to be able to talk me through, if I've given that term, describing where it fits. So if I ask you, what is the somatic motor system? You would say that is an efferent motor pathway that controls voluntary skeletal muscles. Okay? Now, when we look at, again, the peripheral nervous system, you have areas that are coming in versus areas going out. Most of your nerves, so let's say this is cranial or spinal nerve 3. So it's coming out, but in that spinal nerve, there will be axons that are afferent, bringing information from receptors in and around the third vertebral area in your neck. And there will be motor that maybe comes out, and maybe this comes out in cranial nerve 4, or maybe it also comes out in cranial nerve 3. So our nerves in our peripheral nervous system may have axons and fascicles that are collected and made, groups of the afferent nerves that are all collected together and carrying all the sensory information from the pain, the temperature, the stretch, the vibration receptors for that kind of at-level region. And then all of the motor neurons that go out and control the muscles in that region and that area are going to usually be gaggled together in a fascicle. And it might be that those fascicles form one big cranial nerve. So this could all be cranial nerve number 3. Or it could be that some of the sensory coming in at cranial nerve number 3 might affect some of the motor control of muscle cells controlled by neurons in cranial nerve number 4 or cranial nerve number 3 on the opposite end of the spinal cord. Okay? This here is to just kind of show you what a simple reflex, and you do need to be comfortable with the components of a simple reflex, and that'll be later on. Okay? Too fast. All right, so starting with neuro tissue, the functional unit of a neuro tissue is a cell known as the neuron. Okay? The neuron is going to typically have somewhere a nucleus, and the nucleus is typically going to be in a region of the neuron known as the cell body. There will be a part of the neuron that has membranes full of receptor proteins that can respond to a variety of neurotransmitters and neuromodulators. And so that area of the neuron is going to be its receiving end. So it's a one-way communication. It's a one-way street, and the receiving end of the traffic for that street is going to be its dendritic part. And then it's going to have usually many dendrites and then one exiting part that is going to carry the impulse, the electrical signal, down to the terminal site, and that is going to be the axon. Okay? Now, neurons can look like this, and it looks like a little one-eyed monster. And when they look like this with the dendritic parts of the cell coming off of a central cell body, so it kind of looks like hair and a one-eyed monster with one leg, this is known as a multipolar neuron. And multipolar neurons are very common, but they're not the only way that we see neurons. So just be aware that not every neuron looks like this. Okay? But every neuron at some point has a cell body where the nucleus is located. And at some point the cell has an area where it receives information, chemical signals, and that's the dendritic part. And then somewhere it's going to have a part of the body where it's carrying electrical potentials to a terminal site so it can pass the signal on with a chemical release. So it converts an electrical signal to a chemical, and that's the axon. Okay? So this is just, again, getting you comfortable with all of the anatomical terms that you should be able to identify on the model in lab for class, as well as be able to define all of these terms should they show up as a matching activity on the test or fill-in-the-blank activity on a quiz. All right? So again, the parts of a neuron, you have the cell body, the dendrite, and the axon. Those are usually going to be 3 key locations. Where is the nucleus, where is the receiving end, and where is the, the action end, the area where electrical signals are happening or not happening and getting converted to propagate and move the signal onward to another cell. It's still a cell, so there are mitochondria, there's ribosomes, there's rough ER, there's Golgi bodies, there's cytoskeletal proteins, cytoplasm, all right? And then just like in muscle, in the axon, we will actually give some new terms and names to the plasma membrane, the cytoplasm, for specifically the axon of the cell. Okay, so just get yourself comfortable. I want to point out that Nissl bodies is a new term. So unlike a muscle cell that can be really, really, really long, it has multiple nuclei. Well, the neuron can also be extremely long. So if I want to control the muscles in my soleus, the neural cell body and dendrites are sitting in my spinal cord. And so the axon has to leave the spinal cord, travel down my leg, through past the femur, past the quadriceps and the hamstrings, past the knee, all the way down into my soleus gastrocnemius area. So if I need to make any kind of proteins or neurotransmitters, I don't want to wait on the cell body up in my spine to get a message, make it, and then send it down. So I want to have these little kind of like pre-ready factories where I have the ribosome, I have some tRNA, I have some endoplasmic reticulum, I have some amino acids all ready, and the message RNA all ready and packaged together to be a little factory for making a protein that I might need for the physiology of the cell to work and do its job. And I just want to make that into a little grouping, a little like self-making factory. So mRNA, ribosome, RER, transfer RNA, and amino acids. And all of that is going to form this Nissl body, which is then able to stay around the nucleus of the cell, travel down the axon, and just be located in multiple locations. And so that way I don't always have to go back to the nucleus, and especially in long neurons, have a long journey to be able to get any kind of changes to protein synthesis and product making. Okay? All right. Neurons need to communicate to other cells. Sometimes a neuron might physically be touching a cell, but that's not the norm. So muscle, smooth muscle and cardiac muscle touch each other, and what happens in one cell happens to the other because they're physically touching. And in the heart, they actually share cytoplasm. For many of our neurons, they are not physically touching their target. They're not being physically touched by other neurons. So there is always a space, and that space is a synaptic cleft. And so when the electrical signal is traveling on any one given neuron, it can't jump across that space to the other neuron and can propagate that way by jumping across spaces. So the neurons have to take an electrical signal and convert it to a chemical and release the chemical so they can trigger the other cell to either accept that information and continue to propagate the electrical signal, or stop that cell if it has electrical signals happening from continuing to propagate electrical signals and inhibit it. Okay? So when we look at synapses, there's always going to be two cells. One is going to be the neuron, and usually the neuron is going to be the cell bringing electrical signals to its termination area, the axon, and then turning that electrical signal to a chemical by releasing vesicles into the membrane space between the two cells. So that is normally known as your presynaptic cell, your presynaptic neuron, and the membrane that the vesicles will bind to is normally known as your presynaptic membrane. The cleft, the synaptic cleft, is the space between the two cells, and then the cell receiving, which could be a muscle, which could be a gland, or it can be another neuron, is going to be your postsynaptic cell. And the membrane area closest to the presynaptic neuron and presynaptic membrane is going to be your postsynaptic membrane. And for muscle, remember we called the postsynaptic membrane the motor end plate of the sarcolemma. That was the area that the muscle put its acetylcholine receptors in high amounts, so it was increasing the likelihood that if acetylcholine is released from the neuron, it would bind to its receptor. Right? And so you can kind of see in the picture here, this is potentially a neuron talking to another cell. This is the neuron's terminal site of its axon, its teleodendron, and then its synaptic knob. There are lots of proteins in here, these vesicles. It could be that these are neurotransmitters, and the neurotransmitters, when the electrical signal comes down, the action potential, it converts it so that way these vesicles get a signal to merge. As we're seeing here, they merge with the membrane and spit out their contents. And their contents, because the cells are so close and the space is pretty small, are hopefully going to bind and find their receptor. But all of these molecules move by random motion. So some of them might go out. Some of them might be like turning around and try to go back inside the cell. But the reason we release 3 or 4 times the amount we need is so that a large majority of them will, in fact, go and interact with their receptor. Okay? Now, what are these molecules? They're made in a neuron, and they transmit chemically the electrical signal from one neuron to another. So that's why they're called neurotransmitters. So similar to hormones, they can be a variety of background molecules. They can be amino acid derivatives. So norepinephrine and dopamine come from the amino acid tyrosine. So epinephrine is our hormone that comes from tyrosine. Norepinephrine and dopamine are going to be key neurotransmitters that come from a variety of neurons, the sympathetic neurons for norepinephrine, and for dopamine in certain areas of our brain, they play a role, again, Parkinson's disease, in regulating some motor function. So they are amino acid derivatives. All right? Acetylcholine is a parasympathetic neurotransmitter, and it's a little bit more complex, but not that much different from dopamine if you look at it. It has an extra red and an extra black on it. So it's again another amino acid derivative potentially. Another amino acid derivative we can sometimes hear about is tryptophan that gave us melatonin, but it can also give us other neurotransmitters. All right. So some of our neurotransmitters might be proteins. Some might be modified amino acids. Some might be glycoproteins. So they could be, just like hormones, a variety of things. Okay? Now, unlike hormones, they're being released in a small space to try to very quickly interact with their receptors. So they're not trying to travel in the blood a long distance, and they're also not trying to have long-term effects. Most of the time, they're trying to have an immediate effect. So most of the time, neurotransmitter receptors are going to be ion channels. So they bind to their receptor, and upon binding to their receptor, they're going to change ion balance between what's inside the cell and what's outside the cell. And in making ions flow in or out, they are going to change the resting membrane potential. Okay? Now, how do I get rid of neurotransmitters? Because I want neurotransmission to be fast. So if we go back to that previous picture, sometimes I might have a transporter. So that way, as I release neurotransmitter, some of it can be pulled back into the cell and reconstituted into new vesicles. All right? Other times with acetylcholine, I'll have an enzyme in the synapse that can break down the molecule into 2 pieces that no longer are able to bind to the receptor, so acetylcholinesterase. I can also do a variety of other things, take them up in vesicles when they bind to their receptor. There's lots of things. But the key is that neurotransmitters, they are usually something that I can make in large amounts pretty easily. I release them into a synapse, and I release more than I usually need to ensure that some amount of them are able to, through random motion and constant motion, hit the receptor and trigger an ion channel to open, allowing either an ion to flow in, like sodium or chloride or calcium, or allow something to flow out, like potassium. And in doing that, I change membrane potential. And when I change membrane potential on the dendritic surface of a neuron, that might lead to a graded potential. If I change the membrane potential of a muscle as a skeletal muscle or a smooth muscle, I now potentially change muscle through the sarcolemma into the T-tubules and release calcium from the sarcoplasmic reticulum, or in a smooth muscle case, I release calcium and get calmodulin and calcium to then turn on actin-myosin interactions with myosin light chain kinase. Okay? So those are how neurotransmitters work. They have immediate, quick effects. Okay? Whereas hormones, remember, are slower effects because they usually end up changing things or traveling long distances, and so their effects are not within a second or two. Now, neurotransmitter effects will be very quick in turnover, so if I want to, in many cases, keep the effect going, I have to continue to have the neuron stimulating and releasing and forcing an effect, or I need to have, like in norepinephrine's case, a hormone come around and prolong the effects. Okay? Okay. So again, I have lots of different neurotransmitters. You do not have to know any of them by heart or structure. You need to know acetylcholine, norepinephrine, because for the parasympathetic and sympathetic control, those are the key players. Okay? All right. Neurons. So remember earlier I told you not every neuron looks like a multipolar one-eyed monster. Some neurons are going to have different structures depending upon their anatomical location, their function, but in general, all neurons typically have many inputs. They have areas for the dendritic surface to be pretty robust, so they can receive lots of information at any given point in time, and they usually have one part that is an output part, one axon. All right? Now, anaxonic neurons look like they don't have a very distinctive axon. They look like they're mostly a cell that receives a lot of input. All right? So they have a cell body, and then all around it seems to be all this branching. Something on here is an axon, but you can't differentiate it very clearly, so that's part of the reason why they call it anaoxic neuron. These are going to be found mostly in your brain, okay, and some of your sensory organ areas, but again, they just don't need a very long axon. And then they have lots and lots and lots of dendritic surfaces. The bipolar neuron is very rare. You see it, we will see it in the eye in lab, and we'll talk about it in the eye when we get to chapter 17. But the thing about the bipolar neuron is the cell body has basically two projections. One of the projections is tied to some type of receptor, so the photoreceptors or some smell receptors or some hearing receptors, and those receptors then tie into the many dendritic parts of that neuron, and then because it needs to get to the spinal cord or because it needs to travel a little distance, it kind of funnels into what looks like an axon, but it's just a dendritic part of the cell that's carrying action potentials, carrying electrical potentials to the cell body. Now, if this axon is going to carry the signal onward, again, the same thing that would happen on this cell and our multipolar cell happens. The cell body integrates it and takes in information and then determines if it carries it onward. Okay? The unipolar cell, we are going to see these in our reflexes in the spinal cord for sensory input. So these are afferent neurons. Now, they show you that the axonal part that connects the dendritic receptor field and receptor surfaces and the axonal part leaving are equidistant. But in reality, what happens is the dendritic part of the cell is tied to your temperature receptors or your pain receptors or your touch receptors in your skin. And then the axon starts right away and carries that signal all the way to the spinal cord. And it just so happens that the cell body is usually located very close to the spinal cord, and it's a gaggle of many, 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 many of these neuron cell bodies. And together they form what's known as the dorsal root ganglia. And so technically speaking, the amount of axon from the cell body to the synaptic terminal and the spinal cord is usually pretty short, but the axon that comes from the dendritic area to and towards the cell body is usually pretty long. Now, the cell body is like a little leaf. It doesn't actually integrate and take, if there's enough signal happening that the dendritic material responds, the initial segment of the axon is located right here, and so it'll take that information all the way to the terminal site. And so the cell body does a little bit less of a graded potential system there, okay? The graded potentials just occur in this region. All right, and then our multipolar neurons, again, are the most common, and we'll see these with our motor neurons where they are in the spinal cord and embed and then come out and synapse on the muscle, and we're going to see lots of them in interneurons and all over the place. Now, those are the functional cells that are carrying action potentials. But those cells never divide. They are terminally differentiated. So we have to keep them alive. And the way we keep them alive is with these supporting cells, and all of these supporting cells are called neuroglial cells. Now, that's how you learned it in Chapter 4. Now it's time to go that next level. All the neuroglial cells have special names and special looks and special features, and they do different things. 4 of them are unique to the central nervous system, so you will only find them in and around neurons of the brain and spinal cord, and 2 you'll find in the periphery. The names of the 4 in the central nervous system are epidermal cells, which line your ventricles and play a role in making cerebrospinal fluid and trying to keep blood and material that's in the bloodstream and in the extracellular fluid from having access and a barrier to the nerves and the cerebrospinal fluid. Okay? Your astrocytes are also going to wrap around capillaries that kind of push in a little bit more than normal nearest your neurons. They are going to form the blood-brain barrier, and they are also going to wrap around and be other options and functions to help keep neurons alive. Your oligodendrocytes are going to look, I call them like octopuses. All right, they're going to have a central area, and then they're going to have all these arms, and these arms are going to go around and wrap around with a fat-heavy area full of fat, so full of myelin, and they're going to insulate multiple parts of a neuron, multiple parts of maybe 2 or 3 neuron axons, and so they are kind of, like I said, an octopus with their arms wrapped around multiple spots on one axon and maybe multiple spots on a few axons. Okay? And they are found in the brain and spinal cord, and their myelination, their ability to wrap around and insulate, helps the axon have what's known as saltatory propagation, which means it allows it to jump its action potentials longer distances, speeding up the rate at which the action potential gets from its axon hillock initial segment to the terminal. The microglial cells, all right, are going to be a little bit of a neuroglial cell that acts as an immune cell. They're not immune cells, but they are going to basically be your cleanup cells for debris, for damage, for material in the cerebrospinal fluid and in and around the neuroglial cells and the neurons. We don't want macrophages and lymphocytes to be able to get past the blood-brain barrier and into our cerebrospinal fluid, so this population of cells kind of acts for them and takes care of some of the cleanup duties that the neutrophils and the lack of macrophages and T cells has. Now, in the periphery, your myelinating cell is different. It is not going to be like an octopus. It's going to be one little independent crepe, so one little pancake of a cell that wraps around one individual specific segment of one axon. The cell body of that cell is going to be pushed to the outermost area, and that's going to be your Schwann cell. And then your satellite cells are going to be the cells that when we put all of those unipolar cell bodies together in a gaggle, they are going to be the supporting cells that keep those cell bodies alive and form part of like the dorsal root ganglia. They're also going to be some of the supporting cells in and around where synapses are occurring between neurons outside of the central nervous system, so in the sympathetic chain ganglia and some of those areas. Okay? So you need to know all of these cells by name, by location, and by function. What do they do? Okay. Now, the central nervous system, because we see people with spinal cord injuries, strokes, and brain damage, the neurons are very sensitive to stress and change, and those stress and changes can kill them. And when they die in the brain and the spinal cord, they are unlikely to grow back, especially if the cell body of the cell is damaged. However, in the periphery, in many cases, the cells can regenerate their axons, and part of that goes back to the cell bodies embedded in the brain and spinal cord, so as long as the cell body is safe in those regions, the axon, if it does incur damage, can degenerate and kind of get rid of the damaged and the missed parts, and then can regrow itself back to its target because of the Schwann cells having the ability to guide the growth and promote growth of the axon. So that whole process of being able to grow peripheral nerves is huge in that, like, let's say when you have surgery and they go into your knee, it might feel like for the first few months after surgery your entire knee is like numb, and when you touch it you don't feel much and it tingles. And then by the time you're a year out, you might have one or two like little minor dead spots, but for most of the feeling and tension and vibration and pressure, you feel it because it comes back. And so those neurons that were damaged grew back and reconnected to their sensory receptors. Right? And one of the keys in this process of being able to have peripheral nerves regenerate and regrow is that when the axon gets damaged, they go through a process known as Wallerian degeneration. And that is where the axon at the injury site to the receptor are taken out, removed, and then a new axon is grown with the help of the Schwann cells, growing it back to its receptor. Okay. Now we're starting to get to how the neuron works, and this is where we get into electrophysiology. All right, and this is where I asked everyone to please watch the 3 videos listed on our Blackboard page of resting membrane potential, graded potential, and action potential. And it's important you understand this concept because when we get to cardiovascular physiology, we're going to make comparisons between the action potentials in a heart cell, a pacemaker cell, and a ventricle cell back to the neuron. Now, those are the 3 potentials where it comes down to what ion is moving in or out of the cell to what degree with help of what proteins. When that all takes place, that leads to activity in the neuron to be actively either carrying and propagating signals or not, and propagating the signals electrically and then changing that signal to a chemical at the synapse. And then again, when they're receiving a lot of information, integrating all that information that's being generated by receptor ion interactions, receptor neurotransmitter interactions, and receptor ion channel changes, and determining if the signal is strong enough to propagate onward. Okay. So again, I asked you to watch a video. Understanding how all this works comes back to understanding that molecules want to equalize their numbers. So if I have 10 molecules on one side of the cell outside of it and I have 2 inside, those molecules want to try to get 6 and 6. They want to equalize. But because the membrane is not always permeable to those ions, they can't equalize. And then when we take into account that many of the molecules that are small have a charge, they have a positive or negative charge, the molecules, again, if I have 10 positive molecules outside and inside I have a large amount of negative molecules, electrically speaking, those positives want to go inside the cell to be near a large population of negative. So we have chemical gradients that come down to the physical numbers of how many of these molecules I have outside versus inside, and they want to try to equalize versus inside outside. And then I have an electrical component of how many negatives do I have inside, and negatives are fixed more or less because proteins are negative. They tend to have a negativeness to them, DNA, RNA, and those molecules, DNA and RNA, don't usually leave the cell. So we tend to have a desire of positive things, sodium, calcium, magnesium, that would want to, because they're positive charge, come into the cell. And so the resting potential is finding this kind of balance when the cell's not going through any extraordinary measures or triggers or stimuli of what comes in and what comes out, and finding a balance of the chemicals are coming in by chemical means and coming in by electrical means, and it's balanced with the chemicals leaving by chemical means and leaving by electrical means. Okay? And so you need to watch those videos. You need to watch the videos and then start to learn these key terms. All right? Resting membrane potential. And for the neurons, resting membrane potential can be somewhere around negative 70 millivolts. The negative tells you that the negative charge tends to be in the inside of the cell. The 70 kind of gives you an idea of the power between the concentration and electrical gradients for the molecules that are going to try to go in and out. All right? When the resting membrane potential becomes closer to 0 or becomes less negative, we tend to say that is a depolarization event. When the resting membrane potential moves away from threshold, moves away from zero, becomes more negative, we tend to say that is a hyperpolarization event. Whenever I have a depolarization, so I move from my resting membrane potential of -70 to -50. When I go back to -70, I'm not hyperpolarizing there. I'm just returning back to my normal. So that's going to be repolarization. Okay? So no depolarization, repolarization, hyperpolarization. And know that most of these changes occur because neurotransmitters interact with a receptor, and that receptor triggers an opening or a closing of some type of ion channel, allowing some type of chemical and electrical gradient to be engaged where things want to flow in or out. Now, when neurotransmitters open an ion channel, they might only open a few ion channels, and it might be that they make some small change to the resting membrane potential, and like a ripple, it's going to kind of oscillate away from the initial source. If it's not at the axon hillock and it's not causing a very coordinated event, you just call it a graded potential. And so dendrites to the axon hillock, that is where, or the initial segment of the axon, that's where graded potentials exist. And they are small changes in membrane potential, and they might be enough, depending upon how close and how strong the graded potential wave is, they might be strong enough to generate one action potential. But in many cases, it takes multiple graded potentials to kind of summate or add together to create enough strength at the initial segment of the axon to generate an action potential. And in many cases, materials being released to a neuron are generating some graded potentials that are excitatory and some that are inhibitory, and they're canceling each other out. Okay? Now, ion channels are not the only type of channels that we typically have on a membrane. So remember back in hormones, we had receptor membranes that generated a G protein. So those G proteins generated second messengers. Those are other types of chemicals. Or types of receptors and types of proteins, all right? So when we look at some of the membrane channels, the membrane proteins embedded in our neuron, if the neuron channel needs to have the neurotransmitter bound to it, we call that a ligand-gated channel. Okay, and so acetylcholine on the motor end plate binding to its receptor, letting sodium in is a ligand-gated channel. But not every protein embedded in the membrane is a ligand-gated channel. Some of them are going to be what's known as voltage-gated channels. And so they can open or close based on when the membrane potential reaches a certain amount of depolarization or hyperpolarization. And so these are going to be the critical membrane proteins that come into play in electrical potentials, action potentials. And they are very much kind of have a set. They are open maybe from -50 to +30 voltage, and when the voltage is below -50 or above +30, they're not open, they're closed. Okay? And the third type of channel that we can have are mechanically gated channels. And these are the ones that are going to be open because you physically deform the membrane of a cell. So these are some of the channels that we might see on your stretch or push receptors, that if the cell is not manipulated and the membrane isn't pulled or stretched, the membrane channel isn't open. But when you pull or stretch or manipulate the membrane, it makes that channel open. And so that's sometimes how ions can get in. Okay. So again, graded potentials are small changes. So they're not going to be action potentials. They're going to be either a little bit of depolarization, repolarization, or a little bit of hyperpolarization and then repolarization. The goal is that a neurotransmitter is binding to a receptor and triggering the small change in membrane potential. And if the small change in membrane potential is enough at the initial segment of the axon to get it to go into an action potential, that's how we either propagate or inhibit signals on neurons. Okay? For those action potentials, now we're looking at the change that occurs is voltage-gated channels. And that means it's going to be a very uniform, repeatable process because the voltage-gated channels will always open when there is a certain criteria of voltage on the membrane. right? And so in neurons, the action potential is always going to look like this. And it's going to look like this because there is a number of voltage-gated sodium channels sitting at the initial membrane of the axon hillock, initial segment of the axon, that when the membrane potential changes, because graded potentials reach threshold for these voltage-gated channels, threshold being around -50, -55 millivolts, or -60, that means that those voltage-gated sodium channels open. And when they open, they are going to allow sodium ions to flow into the cell in large numbers, because sodium has a concentration, a chemical gradient where there's high sodium out, low sodium in, so it wants to come in high amounts to equalize. And then electrically, sodium is positive and there's lots of negatives in the cell, so electrically it wants to come in. And so that massive influx of sodium is going to mean that from a resting membrane steady state purposes, I'm bringing a lot of positive and I'm bringing a lot of molecule inside the cell. And so the resting membrane potential is going to go to a less negative value, and it's going to continue to rise until the voltage-gated sodium channels close and inactivate. And when they do that, no more sodium can come in. Now, I have voltage-gated potassium channels. Potassium is my positive ion that is in high concentration inside the cell and very low concentration out of the cell. So it has a chemical gradient that when I need to get positive out, I can very quickly open voltage-gated potassium channels, or mechanically gated or ion-gated, and it'll want to leave despite the fact that negative things live inside the cell. Okay? So while electrically it wants to stay, the chemical gradient wants it to leave. So voltage-gated potassium channels open, and I start to let a positive out, and in letting a positive out, I start to get myself back towards resting membrane potential, where the number of negative and positive things inside the cell become more negative in number than positive, and I start to get back to a resting membrane potential. Okay? Now, when I'm looking at an action potential, there's periods of time where to get another action potential to start is virtually impossible. I don't have enough voltage-gated sodium channels that are closed and ready to open, and I don't have the right conditions of ions and chemical and electrical gradients. And that part of time under an action potential where I will not be able to get an action potential to go again, even if I really want it to, is called the absolute refractory period. Now, at some point towards when the action potential is ending, I'm beginning to get the chemical conditions back to where positive and negatives in and outside the cell are going back towards wanting negative to be hoarded inside and wanting potassium to kind of counterbalance that inside the cell, and little potassium out, lots of sodium out, and it's positive and it wants to come in. And so if I need to get another action potential started and I'm not quite finished with the previous, if the stimulus is stronger and more powerful, I could potentially get another action potential to start, is the relative refractory period. Okay? So the takeaway, this action potentials occur on the axon. They occur in a repeated pattern. So every time I get an action potential to happen, it would measure the exact same thing. It would measure for a certain amount of time a depolarization to a certain depolarization value, and then it would repolarize in a certain amount of time, and then it would kind of hyperpolarize because it overshoots a little bit, and then the whole time of the action potential would be exactly a repeatable same feature. All right? And all that comes back to it being voltage gated. If you hit threshold and those voltage gated channels are at threshold, that's where they open, you are getting an all or none response. I then want you to also look at the 4 key phases here of what's going on, and I want you to be able to talk me through. What is it that gets me to threshold? What happens at threshold? What happens at peak? And then how do I get back to resting? Okay? And again, there's a video where, again, following graded potentials and resting membrane potentials, he talks you through all this. All right? Now, action potentials start at the axon hillock, and because you don't really have voltage-gated channels on the cell body and the dendritic part, the action potential is not going to travel towards the cell body and towards the dendrites. It's going to travel towards the axon terminal. And because of the way it starts at this initial segment, it only goes one direction. If I was able to get the axon to start in the middle of the axon, it actually would propagate both directions, towards the cell body and towards the terminating part of the axon, but I never start action potentials in the center, or very rarely. So that's part of the reason why I get one-way communication. Now, how can I get the action potential to travel as fast as possible? Well, I can do a few things to the axon. I can make it pretty big, I can myelinate it, and I can let the axon jump from membrane space exposed. Thus, I'm able to take kind of like big steps. Versus if I don't have myelination and I don't have a very thick or big axon, I have to travel inch by inch, millimeter by millimeter. So the conduction that's the fastest is on the biggest axons that have myelination, so I can jump big distances and I can basically move from axon initial to axon terminating in maybe microseconds. Whereas small, unmyelinated axons, I have to travel every single segment of the axon, and it takes a little bit of time to reset and reestablish. That's going to maybe take me longer to get from point A to point B. Okay? And so the type of fibers we have, the type of neurons that are myelinated and big are called type A fibers, and they can travel 140 meters per second. Okay, I'm about 6 meters tall, maybe, you know, like 5 meters tall, something like that. So if I have axons in my system that let me travel at 140 meters per second, that means I can get information to a point in my toe from my head, you know, in super fast. Right? B fibers are myelinated, but they're smaller in diameter, so therefore they're a little slower. But still, 18 meters per second is pretty fast. C fibers are unmyelinated and small. So again, I might be 5 meters, so in order to have maybe something from my brain to my toe, it would take at 1 meter per second 5 seconds before I would get information from the brain to my toe or toe to my brain. Right? And again, these are just going back to how can I make some neurons fast for moving electrical signals and some slow. Another way I can modulate how neurons work is I could potentially put more voltage-gated channels, more receptors on the neuron. But in order to do that, I would need to manipulate the neuron's physiology, the amount of protein being produced, the amount of maybe sarcolemma proteins and turnover and transporters available. To do that, I want to change the neuron's physiology. So I don't necessarily want to have a synapse. I want to have molecules that, upon acting with the neuron, change its gene expression, change its availability of membrane proteins, change maybe where the membrane proteins are located, so they're closer to the initial segment of the axons, they have more powerful influence. And to do that, I use neuromodulators. Now, some neuromodulators are also neurotransmitters. So dopamine, serotonin are both neurotransmitters and neuromodulators. When they're neurotransmitters, they're going to bind to a receptor that triggers an ion channel and triggers a graded potential on a dendritic surface of the neuron. When they're neuromodulators, they're going to use a different receptor that might have a G protein, like our hormones, that might have a turning on of enzymes inside the neuron that lead to more gene expression of channels and proteins and content on the membrane. And so when you look at neuromodulators, they can potentially make the synaptic communication between two cells increase or decrease in speed and quickness and ability, because they can increase or decrease the vesicles, the neurotransmitters released, and the receptors, and the transport moving, and the acetylcholinesterase, you know, the cleanup molecules. And so that's how they modulate. So opioids are a big category of neuromodulators. They tend to work on pain neurons, so neurons that carry pain action potentials, by making it harder for those neurons to carry pain signals. So the pain signal still happens, it's just you might not get as much of a effect on certain conscious areas of your brain, because the opioids inhibit some pathways for brain pathway information to reach conscious awareness. All right? Cocaine is potentially a neuromodulator because what cocaine does is it makes dopamine transport blocked. So when dopamine is released at the synapse, there's a transporter that tries to put dopamine back into the cell so it can be reused, and so it gets out of the synapse, and so the transmission happens quick and fast, and then it's over. Well, if cocaine blocks that dopamine uptake, the dopamine stays in the synapse and interacts with the receptor, or more likely and for longer periods of time. And so that's why cocaine could be considered a neuromodulator. So again, neuromodulators can be neurotransmitters if they bind to a certain receptor, but in most of the cases, the neuromodulator is altering the neurotransmitter's ability to interact with its receptors. It's altering the cell's neurotransmitter content, the neurotransmitter uptake, the neurotransmitter breakdown. It's altering the receptor numbers, the receptor distribution, and therefore affecting the neuron's ability to react as efficiently or less efficiently to neurotransmitters in a synapse. Okay. So again, when a neuron gets a lot of neurotransmitter binding to its receptors, it has to process that. And then when it processes these graded potentials, again, the graded potentials, if they lead to depolarization events, are considered excitatory graded potentials, and those that are inhibiting and hyperpolarizing and taking the membrane further from threshold at the axon hillock where those voltage-gated sodium channels that are going to trigger an action potential are, we call them inhibitory potentials. All right? And if a lot of our neurotransmitters are causing excitatory potentials, the information processing is probably going to lead to enough graded potentials of excitement that the axon hillock will reach threshold and propagate down an excited message and lead to that neuron releasing neurotransmitter on its next feature or its next cell that it communicates with. If a lot of the neurotransmitters binding to receptors are inhibiting and causing inhibitory graded potentials, then the idea that the axon hillock initial segment will reach threshold and generate an action potential becomes less and less and less likely. And so in a sense, those type of IPSPs and inhibitory graded potentials and hyperpolarization events are going to lead to that neuron not ever wanting or being able to reach threshold at its voltage-gated axon hillock initial segment, and so that in neuron, in effect, is always being inhibited from communicating onward, all right? And so that would be an inhibitory kind of information processing. All right? Normally, again, one graded potential from one neurotransmitter or one synaptic communication from one neuron is not enough to get the axon hillock to reach threshold. So in order to get the axon hillock to reach threshold, it might take more graded potentials from more axons, or from more neurons releasing neurotransmitter from their axon, which is kind of like spatial summation. So again, it would be if I want to get a reaction, I can take 5 or 6 of my fingers and press them in to the body, you know, to say, give me your attention. I'm touching you in 5 different places or 10 different places. And that's spatial summation. I have 5 or 6 different, 10 or 20 different inputs giving on one neuron neurotransmitter release at the same time that trigger all graded potentials that are excitatory, and all those graded potentials and waves coalesce into a bigger tsunami of a wave that definitely gets the action potential started at the axon hillock. Okay. Now, if I only have one neuron, but the one neuron really wants to communicate and get its signal passed on, it can potentially do that by getting successive triggers of neurotransmitter release over and over and over. And so it's like frequency. So it's kind of like if I take my finger and I poke you, and I poke you kind of like once, and then I go, okay, poke you again, okay, poke you again. You might react to that or you might ignore it. But if I go up to you and I go poke, 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 that's temporal summation. I'm trying to get one spot on you, but I'm multiplying it in a very fast frequency to try to get your attention that something important is happening. So spatial summation is all 10 of my fingers push you maybe once. Temporal summation might be one finger pushes you multiple times. Okay? The end result is that a lot of graded potentials are made that summate together that should be big enough of a change on the axon hillock to open the voltage-gated sodium channel. So at this point, that's Chapter 12. That's the takeaways I want you to learn. We'll continue on with Chapter 13 and Chapter 14. I'll try to do another lecture later, maybe today or tomorrow, and get those up. All right, have a great day.